Welcome back to Intro to Physical Anthropology. Gotta remember where I am. Ooh, it's been a long week. I'm David Leitner, I'm your instructor, and today we're going to talk about classification. That is, like, how do you classify life? Like, just what is it? And how do you know when a difference between two species is an important one? How do you know if it's two species or just one? What is a species? These are all questions we're going to answer this week. But to begin with, I really want to sort of focus on classification. So, without further ado, the name for categorizing organisms is called taxonomy. Taxos is categories, and nomi refers to naming things, so it's the naming of categories. And taxonomy can also be the name for the organization itself that you make. Like, you know, the order you put them in is a taxonomy. While you're making it, you are involved in taxonomy. So that's a good word to know. When we're referring to categories in this way, especially in terms of biology, we sometimes refer to categories as taxa. Taxa is any kind of category at any level. It doesn't matter how specific or general it is. The branch of biology that most deals with this stuff is called systematics, and they are most interested in describing organismal variation. However, they tend to use something called phylogenetic classification that I'll talk about in a little bit, rather than the more traditional Linnaean classification. I'm going to talk about the differences a little later in this video. Okay, so let's talk about classifying things with an example. Let's talk about wine. Okay, I'm no connoisseur or anything, but you know, I think it's kind of interesting the way people sort of organize wine or categorize it. You know, we can categorize it by the color, right? Whether it's red or white or rosé, blush, whatever they call it. We can organize them and categorize them by their country of origin or the region of origin, even. We can go even further and talk about terroir, which is a French notion that there's everything from the climate to the composition of the soil to the traditions of how they grow, harvest, and harvest the grapes and make the wine, to even the terrain, the slope, which side of the hill is it on? Like, all of that goes into this, and you can use that to categorize wines as well. Overall, though, all of those classifications have something in common. They are choosing particular traits that seem important and using those traits to classify them. Okay? And it's no different in biological classification. We are looking for similarities and differences in their traits. In systematics, it's this system of comparison is called cladistics, specifically coming from Greek, clados meaning branch or tree-like. And we draw these cladograms, these tree drawings that, like this one here, that help illustrate the evolutionary history and the relationships between different organisms. Organisms that share similar traits usually have a common ancestor. And by looking at the similarities of traits, we can start to figure out how the organisms we're looking at are related to one another. Now, cladograms aren't necessarily very intuitive, so I want to talk a little bit about what you're looking at when you're looking at a cladogram. Cladograms represent a common ancestor and all of its descendants. They are a hypothesis about the connections and the relationships between all those organisms. Typically, the past is on the bottom, the more recent times on top. Sometimes these are read from left to right. Sometimes they're in a circle, so the center is the past and the out. It varies, but they look more or less similar. If you break them down, you'll see it. So you'll have the ancestor in the past there, where you see the little square, and the numbers 1, 2, 3, and 4 are all of the descendants of that ancestor. The tree itself is made of branches and nodes. The nodes mark speciation events. That is, events where a significant change, a split occurred, so that each of the descendants species will go on their own unique evolutionary history from that point on. Branches mark these lineages that link to different species. By the way, it doesn't have to be species. We can talk about any level of organization here. Now, the lineages are really interesting because they reflect evolutionary history. And every species is going to have some part of its lineage that it shares with other species. And depending on what species and how distantly related they are, that shared lineage can be quite long or quite short. But some of it, the last little bit of it, is always going to be unique to that species. All right? So as you can see here, both C and B have their own unique histories after their shared history, right? And A has its unique history after it shared a common ancestor with B and C. Now, B and C didn't magically appear looking like they do right where their histories meet. Those lines there represent lots of intermediate ancestors between them. But they're intermediate, but in a direct line to this particular species we're looking at. The node is simply the point where the shared history ends and the unique histories begin. Each unique lineage has unique ancestors on it. All right? Like I said, all the ancestors on C's unique lineage here, this line, have the same connection to B that C does. Okay? Now a shared lineage, so that's a unique lineage. Unique lineages have unique ancestors. Shared lineages have common ancestors. You think they would say shared, you could say shared, but typically they're called common ancestors. And you can see the yellow and green boxes there represent points where different species had common ancestors here. Green is the common ancestor of B and C. Yellow is the common ancestor of A, B, and C. Now, to make these things, we use the pattern of derived, that is unique traits that evolved in that lineage and nowhere else, and the ancestral or shared traits, the traits it shares with everybody else in its longer lineage. We look at the pattern of derived and ancestral traits so that we can make a hypothesis about how they're related. And in doing so, we try to arrange these groups of descendants into what we call clades. You can see it a little bit here in this chart. This is another way to represent a cladogram. You can see it's not the football goal shape, but it's still kind of generally the same shape, more or less. It's got these branches and nodes. Now here at each node is a common ancestor, and along the lines you have these various traits that developed. And we can group these organisms by how they have those traits. So, for instance, the only trait that fish share in common on this chart with every other animal is having vertebrae. Okay? Amphibians, likewise, although they share vertebrae with fish, the only thing they share with the other animals is having 4 limbs. And then we've got the development of the amniotic sac, and then over here we've got hair and fur develop, and over here we have eggshells develop. And so each of these points here is, a way station between when these features develop. And we can arrange them into buckets. I mean, think of clades as a bucket. You can put everything in the bucket that descended from its common ancestor and the common ancestor. But you'll also notice they're nested. Now, a clade is a monophyletic group consisting of a single common ancestor and all of its descendants. So, if you look at the green square on the screen, that represents a clade because it includes the common ancestor and all of its descendants. If you look at the blue rectangle, that is a clade, even though it only is covering one branch, but it's covering every ancestor in that branch. So it technically is a clade of one. Stop for a second and think if you could tell me why the red square is not a clade. If you said it's not a clade because it doesn't include a common ancestor for every descendant in there, you're correct. There has to be a common ancestor to everybody in the group. And what about the orange square? What do you think? Why do you think that's not a clade? If you said that it's because doesn't include all of the descendants, then you understand clades. That's right. This would be a clade, except it's missing one of the descendants here. So we can't call that a clade. We have to include that other branch to make it a clade. So, the thing about clades is they can be nested within larger clades. So like I said, it's kind of buckets within buckets here. And that hierarchical structure actually helps us understand the evolution of those traits and what order those traits must have evolved in as well. Now, when reading a cladogram or a phylogeny, which is it's like a cladogram with time marked on it, okay? You have to remember a few things. First, the pattern of relationships between species created by evolution is tree-like, not ladder-like. Evolution is not unilinear. It doesn't travel in a straight line. It branches off constantly. We are not the ultimate end of evolution. Evolution has not stopped with us. In millions of years, there will probably be descendants of ours who look very alien to us, may even behave alien to us. They may no longer, large brains may no longer be an advantage in that future. Technology may no longer be an advantage. So they may be very, very different from us. On the other hand, it could be a great advantage. So they could be like magicians or whatever to us. So it's important that you don't read the importance of species based on how far back they go or what order they diverged in. Okay. Similarly, it's not about advancement. We typically also read from left to right when we're reading these things, so that the one that splits off the farthest in the past will tend to be to the left, and the one in the most recent split will be to the right. That's just typically how we do it. You don't have to do it that way, but that's typically how we do it for clarity. But that doesn't imply that one is more advanced than another. Just like I said, we are not the end result of all evolution. We are just one tiny twig on an enormous tree. So, and a twig that's going to eventually sprout new twigs, we hope. So don't read A as being less advanced than B, being less advanced than C, etc., etc. That's important to remember as well. Final thing to remember is the choice of going left or right with a new lineage is entirely arbitrary. It doesn't really matter. What matters is that it's clear enough for you to read and for somebody else to understand. So if you look at the two cladograms below, they are identical from a cladistic perspective. Because it doesn't matter that you can turn around the branches holding the fern, pine, and rose. Look at that. And that's really all they did. They just, they just spun the moss to the other side. That's all they did. But you'll notice you can't just mix them up. You still have to sort of like keep the structure the same. The pattern of branching has to remain the same, whether they go left or right. Okay. So when constructing a cladogram, this is an oversimplified version of this, but this is essentially what the approach scientists take. They start with candidate species that they want to compare on one side, and then they look for traits that they can use to group them together. And usually we're looking at a whole bunch of different traits, not just a few. And not all traits are created equal. Some are better to use than others. But we go through and we list, well, who has vertebrae? Well, frog has vertebrae and 4 limbs. Rodents have vertebrae, 4 limbs, amniotic sac, no eggshells, but hair and fur. Lizards that are, it just goes on and on like that. So you list out the data like that, and then using that as your starting point, you can make the buckets. And the buckets are the traits. So the only trick really is how you arrange the buckets. In order to fit in the bucket, everybody in the bucket has to have that trait, and every trait of every bucket, that bucket is in. So to have hair and fur, you also have to have an amniotic sac, 4 limbs, vertebrae, and be a eukaryote. Right? You have to have all of those. To be a reptile or a bird in this sort of egg group here, you've got to have eggshells, amniotic eggs, 4 limbs, vertebrae, and be a eukaryote. So that's how we draw those. We're essentially looking at them as buckets within buckets. By convention, we tend to put the clade that we're most interested in toward the right, or the most recent clade to the right, depending on what we're talking about. If we want to know just the evolutionary history of this whole group, we try and keep things to the right. If we are just interested, say here in bonobos, we will put the bonobos to the right. Like we could have put the chimpanzees to the right. It would be equally correct to do so. They are equally distantly related from us. They split from each other. Their ancestors that split from each other shared a common ancestor with us. So we come right before them. But you can swing those chimps and bonobos back and forth on that chart, and it's just fine. But say we want to understand bonobos, we'll put them to the right. Now if you add time to a cladogram, some measure of time, instead of just, so far there is sort of a measure of time here. There's a before and an after. We can talk about, gorillas split off before the lineage before humans. Humans split off the lineage before chimps and bonobos, and chimps and bonobos split last. Like, we can talk about before and after, but the distances tell us nothing about actual time passing. It just says before and after. A phylogeny has actual time measurements on it. So, this is not a cladogram, this is a phylogeny, because instead of just reading before and after, we're saying the common ancestor of all of these existed a little after 500 million years ago. That's what we're saying. These are very specific, so that, to be honest, if we were just looking at relationships, this common ancestor and this common ancestor would be on more or less the same level in our representation. But they're not in terms of actual history. This split occurred long before this split did. And so phylogenies give us additional information about the evolutionary history of a group of creatures. We can make comparisons across species and across clades as well. Now, did I misspell Linnaeus? Anyways, the Linnaean system of classification you're probably familiar with from school. Kingdom, phylum, class, order, family, genus, species. It's a nice, neat order. You can slip in some subspecies, subgenus, genera, that sort of thing in there if you need to, but they're kind of all on the same level. Every genus is on the same level, every family is on the same level, etc. Well, the problem is that implying that those ranks are equal doesn't represent, as we just saw, what evolution really looks like. Either it's in terms of its timeline, in terms of the amount of diversity in a particular group, and in terms of the degree of biological difference. Those two orchids can hybridize, even though they're in different genera. That's plural for a genus. Those cats also are from different genera, so in terms of the Linnaean system, they're kind of equally distantly related, right? Wrong. They are so biologically different, they do not hybridize. I'm glad I didn't run that experiment. A phylogenetic classification system, on the other hand, only names clades. There's no notion of levels here, other than the fact that clades can sit within other clades. That's the only thing. So a clade is itself, right? And that allows us more nuanced understanding of the evolutionary history of a group of animals, or plants, of organisms. In a way, the Linnaean system is a little bit like a bookcase, and you run into problems when you got something that's not in between a species that sits on the in between a group that sits on the family level and the order level. Well, you got to come up with an infra-order now and put it in there, and you're constantly inserting subcategories in there to maintain that hierarchy. But phylogenetic classification is a lot more like Tupperware. You can stack it, you can fit it together, you can make it work. You just throw them all in the bucket and arrange them the way you need to make sense of it. Okay? It's not a great analogy, but I just thought it was funny. So phylogenetic naming systems name every clade in a timeline, or can name every clade in a timeline. This is too hard in the Linnaean system, but as you can see here, every single clade here has its own name. We can talk about Crocodilomorpha, Squamata, Testudines, and Archaeosauria. Archosauria. Sorry, gosh, I gotta work on my pronunciations. However, when you try and insert other kinds of categories, like reptile, this is a Linnaean category, right? Reptile. Well, problem is, the Linnaean categories also distinguish birds from everything else. So this doesn't tell, having birds separate from reptiles, when in fact birds and all the other things we call reptiles share common ancestors, is a little weird. It's just a little weird. On the other hand, using clade names, we can see that actually birds are dinosaurs. They share a common ancestor with non-avian dinosaurs. All of the kinds of non-avian dinosaurs, by the way. There are lots. Incredibly successful group of animals. So, now in classification, when we're looking for how things are related, we are looking for shared derived traits. So a derived trait is a trait that evolves after the last common ancestor with another species. And a shared derived trait is a set of traits you share with every organism in your lineage back to wherever you're drawing the common ancestor with another lineage. Okay? So we look for shared derived traits, just like in the buckets, right? If you all got 4 legs, you all go in the same bucket, but within that bucket, you can have a bucket for amniotic eggs, right? And so forth. Now, should be pretty straightforward, but every once in a while, we have to bear in mind that there are 2 different reasons a trait might have developed. The first is called homology, and homologous traits are similar features in 2 related organisms that look alike because they share a common ancestor. In other words, the common ancestor had that trait too. So they share the trait because they share a common ancestor. If you and your siblings have purple eyes and your grandfather had purple eyes too, that's because, and your dad had purple eyes, that's because you all share common ancestors, right? An analogous trait is one that is similar in 2 related organisms because of convergent, sometimes called parallel, evolution. That means they don't share a common ancestor with that trait. They evolve that trait separately. And I'll show you an example now. This is an example of a homologous trait among tetrapods. So this is the development of the 4-limb structure we see on every animal, well, every tetrapod. That goes back hundreds of millions of years in evolution. And one thing you'll notice is each of these limbs performs a different task. The human limb uses its arm for lifting, for throwing, for gesturing, all of these strange things. Alligators use theirs for swimming and sort of waddling on land. Penguins use theirs for, not for flight, but for swimming. And bats use theirs for flight. All 4 of these are very different functions, but it has the same structure because they all share a common ancestor that had this limb structure. They all have a humerus attached to a radius and ulna, attached to carpals, attached to metacarpals, and at the very end, phalanges. They may be very different shapes, but they all have the same underlying structure because they shared a common ancestor with that same structure. These, on the other hand, are examples of analogous traits. All 4 of these structures do the exact same thing. They produce flight, but they do it very differently. The pteranodon down here uses skin stretched between its, sorry, that's the bat. The bat down here uses skin stretched between each of its phalanges except for its thumb, and between the last phalange and its body. Okay, that's how it creates an airfoil. It can actually control the shape of the airfoil that way. Birds, on the other hand, have almost completely lost their phalanges, as well as their carpals and metacarpals, and instead they've got feathers. These structures that are sort of specialized hair or specialized scales that produce lift and create an airfoil surface. The pteranodon, kind of like the bat, stretches skin, but not from every phalange, just from the last one here, to its body. So it can't control the shape of the airfoil as much as a bat does, but it still does fine flying and gliding. Finally, the last one, these all use a 4-limb. These are all tetrapods, these 3. This is not a tetrapod. This is an insect. They have its connection to the others is much deeper in the evolutionary past. And it is using a completely different body part to evolve this. Wings maybe evolved from specialized shells, carapace, which is kind of interesting. And it still produces flight. It produces flight in a completely different way than any of these others. It doesn't actually create an airfoil shape. It just pushes air around to be able to fly. These are analogous traits. None of these four have an ancestor that had the same trait of flight with the same structure. All of these three share a tetrapod ancestor, an ancestor that had the same sort of four-limb structure, but in a different form. But that organism did not use it for flight. So we cannot say these all evolved from the same common ancestor. These traits evolved from the same common ancestor. So that's really interesting. So that's one of the difficulties when you're sort of trying to classify things, is determining which explanation makes more sense. Now, I said before, I want to leave you with this. I said before that cladograms and phylogenies are essentially hypotheses. They are a prospective explanation for the evolutionary history of something, and that explains the pattern we see in the data. And as you've seen, you could arrange the boxes a little differently if you looked at different traits. And so when we do this, we have to think about when we make a cladogram, we have to think about which boxes are the important boxes, and whether some boxes are the result of homologous traits or analogous traits. Are these separate incidents of evolution? That changes the shape of our cladogram. And how do we tell the difference, though? There's a principle called parsimony. 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 And it's based on something called Occam's razor. Occam. Occam. Occam was a philosopher, medieval philosopher, who basically held that all other things being equal, given two equally likely outcomes, the explanation, or two equally likely explanations, the explanation that makes the fewest assumptions is the most likely to be right. Now, it's not 100%, but it is generally a good principle, and a great principle for critical thinking, where a lot of conspiracy theories and a lot of theories about ancient aliens and that sort of stuff, where they fall down is they have to make a number of assumptions about the existence of things we have no evidence for the existence of. And that is a huge assumption, right? Which automatically handicaps it in terms of some scientific explanations. So this notion of parsimony is the one we use to determine which are the most likely cladograms. Now, again, they're not 100%. Nature doesn't always work that way, but it works often enough that it's a good rule of thumb to go by. And frankly, it's a good rule of thumb to go by in life when people are trying to sell you a bill of goods. Okay? So keep that in mind. Meanwhile, have a great day or whatever. I wish you all the best and take care. See you next time.